Science, create a scientific outlook and organize lectures and seminars. Since last 54 years, every Wednesday and July 1 month at 6 o'clock, talks are conducted on interesting topics delivered by various eminent scholars. Today, 2715 talk on tracking the objects of insects, affection across species and continent is going to be delivered by Dr. Shannon Olsson, Professor, National Center for Biological Sciences. On behalf of Bangalore Science Forum and all of us here, it gives me immense pleasure and honor to welcome you, madam. Dr. Shannon heads the naturalist-inspired chemical ecology group at the National Center for Biological Sciences, Tata Institute of Fundamental Research in Bangalore. The NICE group studies how animals and especially insects make decisions in the nature. Dr. Olsson completed her PhD at Cornell University in neurobiology and behavior and chemical ecology. She then worked at the Max Planck Institute for Chemical Ecology in Jena, Germany as a project leader, where she collaborated with engineers to develop an artificial chemical communication system based on insect chemical communication. A Fulbright scholar and Ramanujan fellow, Dr. Olsson is a part of large effort to promote chemical ecology in India. Her favorite thing about being a scientist is the privilege to observe and learn every day something completely new, and the possibility that the something new could make a positive difference in this world. With this brief introduction, once again I extend a very warm welcome to Dr. Shannon.
Good evening, everyone. It's a great honor to be the 2,715th speaker at this really prestigious event. So quite a number of shoulders, of giants I have to stand on, I think. But it's really quite an honor, and it's really a privilege. So thank you very much. I have to start by introducing the shoulders that I stand upon every day. It's the group I work with in Bangalore here at NCBS. This is my group. We are called the NICE group, Naturalist Inspired Chemical Ecology, and I have a really wonderful group of bright people who are passionate and enthusiastic and constantly challenge each other and myself every single day. And this is really their work that I'm talking about. So I speak upon their behalf, and it's an honor to do so. So I want to start by taking a step back and thinking about biology. Biology is a study of life, right? And if you think about life, all life on this planet, if you're a bacteria, or you're a plant, or you're an elephant, or you're a human, right? There are basically three things that you need to do, right? First of all, you need to reproduce. You can reproduce by yourself if you're asexual, but otherwise you have to, as Queen once wrote in their lyrics, you have to find somebody to love, right? That's a very important aspect of life, and arguably the most important aspect. Another thing you have to do is stay alive until you find somebody to love, which means that you have to find resources. You have to find food of some sort. And you have to avoid getting eaten at the same time. So these are the three things. You need to love, you need to find food, and you need to avoid enemies. And in order to do any of these things, you have to know what you're looking for, right? You certainly don't want to eat what's going to kill you, obviously. And you don't want to run away from something that you're supposed to love. So you have to be able to identify the right objects. The right object is the person you're going to love, a good woman, if you're a man, right? You have to find a good food source, and you have to avoid something like a poison or a tiger, right? All of these things are very, very important. But actually how we as animals, or even the tiniest animals among us, identify these things in their environment is a really complex thought, actually. actually. And to give you an example, I'm going to ask the audience a question right now. I'm going to give you a little quiz, and you can just shout it out, okay? What are these things up here on the screen?
Flowers.
Very good. You all passed the test. Yes, they are flowers, of course. Obviously, you all know this. And you may have your own language, which may or may not be Kannada, for these different flowers, right? But you can identify them all as flowers because you've learned that all of these things are flowers. But if you look at them, they're extremely different from each other, right? They're very different shapes. They're different colors. They're different contrasts. They have different sizes even. And of course, if you were able to smell them, they'd have different smells. But yet our brains are able to categorize them all as flowers, which is quite remarkable. Because if I ask you to break this down and identify what actually makes it a flower, it's a very difficult task because it's not a single color or a single shape or a single smell that defines the flower, but it's rather a group of characteristics that are quite difficult to label, but yet your brain did automatically and instantaneously. And then you take something like this, right? This little tiny fly. This is a hoverfly. This is a pollinating insect. It's a type of wild pollinator. It's one of the most prolific wild pollinators on the planet. And this little fly feeds from the nectar of pollen on flowers and by way pollinates those flowers. And it has to find flowers just like you identified a flower. But yet it's got this tiny, tiny little brain. Its brain is only about 0.5 millimeter in size. But yet it doesn't try to pollinate a rock. It doesn't try to pollinate the grass. It goes to flowers. And in this case, it didn't have mommies and daddies around to teach it. Most insects are actually solitary, which means when they emerge from their pupa or emerge from their eggs, there's nobody there. There's nobody there to teach them what things are. There's nobody to say, look, this is a flower, this is a flower, this is jasmine, this is rose. Nobody. They have to either figure it out on their own, or alternatively, they have to have some innate ability, meaning they're born with the ability to identify flowers. And since I just showed you this slide, you will, I hope, appreciate how amazing this actually is. Because identifying all of these as a flower and actually being born with a sense of that is quite remarkable. So this is really what we study, is how animals, and especially these little insects with these tiny brains, know what things are. They know who to love, what to eat, and how not to get eaten. This is basically what we study. And they do this in an incredibly complex world. This is the Western Ghats where we do a lot of our research. And look at it. Imagine trying to find flowers in this region. Well, I think you can see those red flowers right there. But being able to find things is a very difficult task because there's many, many, many things out there in the world, particularly here in India, where there's a massive amount of biodiversity. So how we are able to identify these objects in the group of objects is quite remarkable. So to start with how we understand how insects identify objects, I wanted to give you this object, which is one of my favorite objects. It's a cup of coffee. And it's one of my favorite things. And I don't go a day without a cup of coffee in the morning. And I first learned coffee by its smell, because my parents brewed coffee every morning. And it was the first thing I woke up to. I woke up and I could smell the coffee wafting through the house. And somebody else who really loves coffee is this guy. This is the coffee white stem borer. This is a massive problem in India right now. It's devastating the coffee plantations throughout both the southern India, but also as well as in the north of coffee plantations. Because what it does is, once the female and the male mate, the female lays her eggs in the coffee stem, and the larvae eat the wood of the stem, and they eat right through it, and they basically cut off the circulation of the plant, and the plant dies. And some of these plants, as you know, can grow to 50 to 100 years old, and just a small infestation can completely destroy the plant. So it's caused really millions of rupees of damage, and it's still a big problem. So, of course, the question is, how does the coffee borer know what coffee is, right? How do they know what a coffee plant is? How do they identify a coffee plant? Now, of course, we take it for granted. You know, if you ask somebody, well, they're coffee borers, so of course they know what coffee is. Yes, obviously, that's true. But what is it about the coffee plant that they use to identify it? And so that's what the students in my group and I set out to do. So we went to Cork, we went to these beautiful coffee plantations, and we set up a field lab, because where better to study how the coffee borer likes coffee than in the coffee plantations, where you can watch them doing their job, finding their host plant in this case. So we built these large cages, which were about 4 by 4 by 3 meters in size, and we released them on Arabica plants, which is one of the preferred varieties of coffee. And we watched them from 10 to 4 in the afternoon, which is usually when they're primarily active. And over the course of the time, the students observed over 795 beetles, right? And the first thing that they did was they wanted to know, where do they mate, right? It's the females that lay the eggs. But of course, before they lay the eggs, they have to mate. So do they mate on the plant? So maybe they find the plant first to mate, and then they lay the eggs. So that was our first question, and they just watched them. They just watched where they went inside their cages. And they noticed that some of them mated on the net, some of them mated where they were released, which is on a pole, basically, and only a few of them mated on the plant, which suggested to us that at least they don't need the plant to mate. They could mate on the plant, or they could not mate on the plant, but they don't have to mate on the plant. So that means coffee is not to them something to love, right? That's not where they find somebody to love necessarily. So then we wanted to know, well, how do they get to the plant? Well, about 95% of them flew. So this means that when they are choosing what to go to, what to eat in this case, or at least what to feed their babies, they flew. And this is important information, because that will give them different information than if they were walking on the ground. And then, of course, we wanted to know, where do they go? So there's many aspects of the plant that they could land on. They could land on the leaves, they could land on the stem, they could land right on the main stem of the plant as well. But 97% of them landed on the leaves, which suggests that the leaves are important. However, there are just a lot of leaves on a coffee plant, so it kind of is logical that they would land on the leaves, because there's a lot of them. So my students took all the leaves off one of the plants. So you see there on the left, you see this poor, sad-looking thing that has been completely defoliated, and there's a plant on the right. And they said, well, if the leaves are important, then the animals should land on the plant with the leaves, and they should avoid the one without leaves. And that's what they did, which suggests to us that the leaves are an important factor in them identifying the coffee object. And then they wanted to find out, what is it about the leaves that is important? So they covered 2 plants with plastic. And this allowed the visual cues to get through. The color was still there, the shininess of the leaves could still be detected, but the smell was gone, right? There's no smell because it's completely covered up by plastic. And in one of the plants on the right, or on the left, depending on the day, they poked holes in the plastic. So that allowed the smell of the coffee plant to seep through. And then they looked at where the beetles went. And the beetles mostly went to the plant with holes, which suggests to us that it's not only the look of the plant, but very much the smell of the plant that is important. So this is giving us a lot more information on what a coffee plant is to these beetles. A coffee plant is something with leaves that have this particular smell. And that's a lot more than we knew when we started the project. So then, of course, we have to identify what those chemical cues are. And this makes a lot of sense, because insects are extremely olfactory. We often talk about dogs being really great smellers. We use dogs a lot to detect bombs. We use them for drug detection. We use them in all sorts of search and rescue operations. They're about 1,000 times more sensitive than humans. But insects are 10,000 times more sensitive than a dog, which means they are really, really good at smelling. They have really pretty bad eyesight because they're so tiny. You actually have to be a certain size to have good eyes. So although they have very fast eyes, their compound eyes make them very fast. As you know, if you've ever tried to swat a mosquito, they aren't very good at acuity. So they can only see things as a blur unless they're very close to it. To make up for that, they smell really well. So we collected the smells of the plant. So this is one of the ways we do it. We just bag some leaves on the left, and we have basically an airflow going through it to some sort of filter. And the filter absorbs the smells onto it, and then we can rinse it with a solvent and identify it with chromatography. But I show you this picture because on the right is our lab assistant who did not cooperate and took our equipment tree with it. So sometimes this doesn't always work very well in the field, but we still try to do it anyways. Another day, our entire lab site was trampled on by elephants in musk in Cork, so you have to be very flexible when you're doing field research. Then once we've identified the chemicals, what we can do is present them to the olfactory system of the insect, which happens to be their antenna. The antenna of the insect is their nose, and this is how they detect volatiles. And here you can see an electron micrograph of the borer antenna on the left, and on the right you can see these tiny little hairs. Those hairs each contain neurons, and those neurons have particular proteins on them that physically bind to the different chemicals. So each hair has its own specific repertoire of things it can smell. And we can identify what it smells by measuring the electrical activity. So what you see on the bottom is the electrical activity of the antenna as we pass chemicals over it. The chemicals there are shown in red. This is actually a gas chromatographic spectrum. And what we have in our lab is we have something called GCEAD, where we can pass the chemicals over the antenna as they pass through the chromatograph, and we can pass them over the antenna and then record the electrical activity at the same time. So this will give us some idea of what it is in the coffee that the insects can smell. And then of course we have to do then behavior to identify what are the actual chemicals that they like about coffee. So this is how we identify objects. But I want to take you back to this little hoverfly. This is a really lovely little creature. There's almost 6,000 species of hoverflies across the globe. And they're really good at finding flowers, in fact. They are so good that we have found them, the same species, here in Bangalore. We found it in Lachen. This particular picture was taken at 2,500 meters, but we even found them at over 4,000 meters. In Jena, Germany, where I used to be a project leader, and even as high up as Uppsala, Sweden, which is the subarctic, way up in northern Europe. This is remarkable, right? It's remarkable that they can find flowers in all of these different climates. Obviously, the flowers that they find in these areas are quite different, which then begs the question, well, we know they can find flowers, but then what is a flower to them when the flowers are so different across these many, many different climates? So then we say, what is an object on different continents? So what is a flower? Well, this is obviously a flower. This is an evening primrose, and you and I would recognize this is a flower as you did earlier. This is also that same flower. This is the ultraviolet spectrum of the flower. As many of you know, insects can detect ultraviolet light, and many flowers have specific ultraviolet patterning that allows insects to identify where is the nectar and pollen. You can see it looks like a bullseye. It tells the insect exactly where to go to find that nectar and pollen. So to an insect, this is also that flower. This is also a flower. This is the humidity spectrum of that flower. Humidity is a very good indicator of nectar, right? Nectar is a liquid. And so when you have a very humid environment, that gives you a good idea, at least if you're a hoverfly, that you're going to find a lot of nectar there. So humidity is also a way of identifying flowers. And as I was telling you a little bit earlier, this is also a flower. This is the volatiles of that evening primrose. So at any one point in time, all of these cues are available to these hoverflies to identify flowers. So here's the question. What do they do? Do they go to all flowers that have this chemical, regardless of where they are? Maybe they just go to yellow flowers. Maybe they go to yellow flowers in Sweden and white flowers in the Himalayas. We don't know. What is it? What are the cues? Are they the same cues in every environment? Or does each environment have a novel group of cues? So to do this, of course, we have to go to each of these places. We have to travel to the Himalayas, and we also have to travel all the way up to Sweden and watch them, right? So we watch these hoverflies in each of these different environments, and we watch what are the flowers that they go to. What are the flowers that they really liked to go to, and what are the flowers that they always stayed away from? And we would observe them for some period of time, and we would go to several different fields and look at them and find them. And once we saw them travel to a flower, like we did see here, we saw them visiting different flowers. You will recognize the ixora, also the rapeseed in Sikkim, and also some thistle in Uppsala. And they went to all of these different flowers. And as soon as we saw them do that, we would then collect the cues coming from those flowers. So we took pictures of the flowers. We measured the color spectrum in both UV and visible light, so the wavelengths that were being emitted from those flowers are reflected, I should say. We also measured abiotic cues, so we measured the humidity and the carbon dioxide and the temperature of the flower. And we also measured the chemical cues using a tiny adsorbent silicon fiber that allowed us to measure what the smells were of that flower. And then we got a lot of data. We got a lot of different smells, a lot of pictures. We got humidity measurements. We got site measurements. We also took panoramas around the flower to get the idea of the environment in which the flower was placed. And this was work done by Pragadish and Shuridan in our group. And then we put it in a giant statistical soup, basically, this sort of big data analysis that we hear a lot about in science now. And this was done by Josephine Dahlbom, who's a postdoc at Uppsala in Sweden. And what she did was she put all of this information together, and then she basically asked. She asked her statistical analyses, all right, what's a good flower if you're in Uppsala? What are the things it should have? What color should it have, or smell should it have, or should it be humid, or should it be round, or should it be tall? She just asked, what parameters are attractive to a hoverfly in Sweden, or in Bangalore, or in the Himalayas? And what characteristics do they really seem to avoid in all of these places? And although we got a lot of statistical data, this data was actually characteristics of flowers. So that means that we were able to create models of flowers. Now, these are not real flowers, right? There's no flower that looks exactly like this or has this particular smell. What this is a result of our statistical modeling of the ideal objects that each of these regions should have to attract hoverflies. So in Sikkim, for example, to our eyes anyways, the flower should look yellow. It should have a wavelength around 400, which is yellow to our eyes. And in Uppsala, it should be white. And in Bangalore, it should be blue. And they should really hate pink flowers in the Himalayas. And they should really not like them when they're shaped like a funnel. So this is all the information that we came up with based upon this multivariate analysis. And then, of course, the proof in the pudding comes to try to make these flowers for real. So making these artificial models is quite a task because we don't have a real flower to predict. So we're not trying to make a rose or a lily or an orchid. We're trying to make some statistical model. So what we did was rather than trying to make a rose or a buttercup or whatever it is, we actually tried to replicate the cues as close as possible. So make the wavelengths exactly the color of flowers. Make the smell exactly what we needed the flowers to smell like, and no stronger than a flower. A strong smell would be like a lady with too much perfume. We didn't want to do that. And we then had to measure them, just like we did with the actual flowers. So taking pictures, taking carbon dioxide measurements, measure it just like we would have done a real flower, and see if it replicates our cues with what we want. Then we took it to the field. So this is one of our field sites. This is the Tanggu Valley. This is about 3,900 meters. This is a lovely nomadic yak village up in North Sikkim. It's beautiful. And you can see these primulas and buttercups. So in our analyses, they loved the buttercups and hated these primulas. And that may be part of the reason why the flowers that they liked are yellow to our eyes, and the flowers that they didn't like are sort of pinky purple. We're not sure. And then we tested the flowers. So we made these fake flowers. And you can see in the left image here, this is actually a bunch of flowers. They're all arranged within also real flowers. We also arranged our flowers where there were no flowers. I'll give you a little clue if you want to see where they are. I've now circled them in red. Can you see that? Yeah. So I think you can definitely see the green one. That looks really, really strange, right? And the pink one. But some of them are a little bit hard to distinguish. Up in the top right, those little white flowers there are actually our replicated flowers. And down at the bottom, you'll see on the far left the green flower that looks really different. But there's also one of our yellow flowers, which is the second one from the right. So even though we weren't trying to make them look like flowers, actually in the middle of our field, they sort of did look like flowers. So we put them where there were flowers and put them where there weren't flowers to see if they were able to attract any flies. And they did. to our flowers. In fact, they really liked our positive control, which I didn't tell you about before, but that was actually where we actually tried to make a flower. We tried to replicate a buttercup using the same techniques that we were doing for the other models, but this time we actually were trying to make a flower. And they also seemed to really like the Sikkim flower, which makes sense because we were in Sikkim, and they also seemed to like the Swedish flower, which is interesting because we're not in Sweden. So now, in about 1 week, I will be able to tell you what happened in Sweden, but the season is just starting, so I can't tell you right now. And then the Bangalore season starts right after monsoon, so I can't tell you that either. So I'm sorry this is a to be continued ending, but you can see that we were at least able to somewhat predict what they would like in these different places. Now the question is, what do they like in these other regions? And is the flower the same thing across all different environments? Next thing we're going, which is where we want to go next, is to make flowers that are a bit more sturdy. So a high school student from here in Bangalore actually got very excited about the idea of 3D printing. And she has designed 3D printed flowers, which I have up here on the podium. So after the talk, you are welcome to come see them. They are really beautiful. So this is a way that we want to make more sturdy flowers to be able to manipulate the cues a bit more easily. Because with paper flowers, it's a little bit like origami. They're not so sturdy. So this is where we're going next with our information. And we can put the smells right inside. You can see there's these little canisters underneath the flower where we can actually have the smells impregnated into some sort of release. All right, so that's that story. So now I've talked a little bit about how objects are in different places in the world. Now there's another very important thing that happens, which is what happens when a new object comes to town. So this is a blue banana, right? Yeah, blue banana. Maybe. It could be an ultraviolet banana. It could be just a drawing that sort of looks like a banana. Let's say it's a blue banana. All right, so raise your hand if you would eat this banana. Some people maybe would try it. So I'm sure if you were desperate enough, you'd probably eat it if you were really hungry. But that's an interesting question, right? Because of course, bananas have many different colors. Some bananas are purple, some bananas are green, some bananas are yellow, some bananas are speckled with brown. There can be many different colors of a banana. And food can also be blue. Blueberries are blue. So there's lots of different things that are these. So it's not that this can't be edible. It's that the combination of cues is something that we don't recognize, at least we haven't learned to recognize as something that's edible. So we're not really sure what to do with it. And this is actually happening all the time in our natural world, particularly here in India, where we have tons of invasive species coming at us all the time. And the question is, if you're an animal or an insect and you have this new thing come into your environment, what do you do with it? How do you recognize it? Because it's completely new. And as I mentioned, you may or may not have the processing ability in your brain to be able to deal with it. Maybe you do utilize it, maybe you don't. It's a really interesting question of what animals do when something new comes into their world. So for this question, I'm going to take you all the way back to my homeland in the United States, to South Bend, Indiana, where my collaborator works at the University of Notre Dame. And we are working with this insect. This is a really lovely insect, and this is called the apple maggot fly. It's called the apple maggot fly. You'll probably figure out why. Because it lays its eggs in apples, and its larvae feed on apples. And the reason that they are so interesting is, in fact, apples are not native to North America. They are, in fact, also an invasive species. They were brought by the colonists when the colonists came to the New World about 300 to 400 years ago. And there weren't very many apples for a long time, but of course, as the pioneers came across America, and especially as they traveled across Indiana, they planted thousands and thousands and thousands of apple trees. So suddenly there was just this massive new object that was never there before. And the question is, how did this apple fly become an apple fly? Because there were no apples before. So actually, Benjamin Walsh first discovered this in 1867. He was actually a friend of Charles Darwin's, and he moved to America because he wanted to retire from science and be a farmer, but he didn't succeed very well, so he decided to become an entomologist instead. And he was Hudson Valley, which is in New York State, and he was just watching them, and he noticed that some of them were feeding on apples, which was weird to him because they weren't apple flies at that time. He knew them as hawthorn flies. Hawthorns are a tall, deciduous tree that produces little red berries, very, very different than apples. So he wanted to know how this happened. So this was about 150 years ago, and I can tell you we are still wondering, but we have some ideas right now, and one of that is that even though the apple is a new thing, actually, if you look at what they like about apples, it's actually the smell. I told you insects are very good at smelling, so smell is a very important factor in identifying objects. And the smell of the apple, although it's unique, and it doesn't smell exactly like hawthorns, these flies were very interested in a lot of different types of hawthorns. And although no particular hawthorn berry smelled like an apple, parts of the smell was similar to an apple. So the apple became this new object that had parts of it from all of these other objects that they already fed on. So it was kind of like a blue banana, right? Looks like a banana, but it's blue like a blueberry. So it was a mixture of many different things. And their system was able to identify it as a potential host source. So they went over to it. And they really liked the smell of apples. So this is a preference test where we put them in something called a flight tunnel, where we release them on one end, and we have the odor of something like an apple, or we can use an actual apple on the other end, and we see do they fly to it. And 80% of apple flies go to the smell of apples, but only about 20% go to hawthorns. So they're not hawthorn flies anymore. They are definitely apple flies now. And the interesting thing is, not only do they not go to hawthorns, but they're actually repelled when you add the smell of hawthorn to the apple. So this is kind of amazing because in 1867, which is about 160 years ago, they were hawthorn flies. There was no apples around. So the fact that they have not only started to like something but hate what they used to love is really very fast in evolutionary time. So we wanted to know, what is it? What makes them love apples, love this object, and hate something that they used to love? So to do that, you have to look into their minds, right? You have to look at their heads. And the most logical place to start is actually not in their brain, but actually on their antenna. Because, obviously, if you become blind to something, or in this case, if you become smell blind, or anosmic, then you can't identify it anymore. So perhaps they don't smell apples anymore, right? Or smell hawthorns. They just have generated new ability to smell apples, and they stopped smelling hawthorns. But that doesn't seem to be the case. Both my own work and my student Cheyenne, who I share with the University of Notre Dame, have found that they all have the same ability to smell. Apple flies can smell hawthorns, and hawthorns can smell apples, and even blueberry flies, who I didn't even tell you about, but are another species. They can smell apples and hawthorns. So it's not because they've just suddenly turned off their ability to sense it. So that means it has to be something that has gone on in their brain. This is what the brain of these apple flies looks like. This is a confocal micrograph of the brain that is stained with different dyes to identify the brain regions. Flies have brain regions just like humans do. They have visual centers, they have olfactory centers, they have memory centers, they have centers that allow them to walk and to have coordination. So they have all of the same types of basic centers of their brain as the mammalian brain does. And so we looked at their brains, we reconstructed them to get an idea of these different brain regions. This is a focus on the olfactory centers, so the centers that code the smell and the processing of odors. And then we did something called optical imaging. So I think some of you might have heard of fMRI, functional magnetic resonance imaging. This is a technique used in medicine to identify brain activity while humans are thinking of something. So you can, for example, play a sad movie, and then you can watch where the activity in the brain is happening, and you can see, this center of the brain is firing when they have this, or you can have them think of a house and see what areas of the brains light up. We basically do the same thing, but we can't do fMRI on insects because the whole brain fits into one pixel of an fMRI. So we have to use a different technique, which we use a calcium dye. Calcium is a very important chemical in the brain. Calcium is exchanged whenever there's a synapse and whenever there's a synapse, there's an exchange of information. So we can basically figure out what the flies are thinking by looking at where there's changes in calcium. So that's what we've done here. So we present these different flies with smells of hawthorn and smells of apple, and we map them onto the brain. And I hope you can see it. I think it shows up. That the hawthorn flies, the green region, which is the smell of apples, is higher in the brain than the red region. And then the apple flies, the red region is higher than the green region. You see that? Okay, so what this actually shows us is that where hawthorn flies think about apples is where apple flies think about hawthorns. So they've reversed. So if you're thinking in one region and this tells you it's good, and then you make it where it's bad, then you've changed your preference. So good becomes bad and bad becomes good, so they've just reversed it. So it's a very simple way of changing an object, right? You just change what the object means. It used to mean that it was bad, and now it means it was good. So that's what we think is happening and how it can happen so rapidly. So these apple flies, when they were going to seeing these new apples, some of them changed their mind, basically. They just changed their mind. Instead of thinking it was bad, they now thought it was good. So we think it was a modulation in wiring that allowed them to identify these cues. Now, one thing that I haven't talked about up to now is actually how insects actually find these things. I talked about how we know what those things are. I talked about coffee. I talked about how these objects change in different environments, or I talked about how animals deal with objects that are new to an environment. But another question we have is, how do they even find them? How do they know where to go? For example, if you're a hawthorn fly, as shown here, this particular behavioral ecologist, who's very well known in the field of entomology, Ron Prokope, he said, well, they go to a group of trees, and then they find out a single tree, and then they go to the group of fruit, and then they land on the fruit, and that's where they lay the eggs. But obviously they have to go to the tree before they go to the fruit. That's pretty logical, right? But the question is, does the fruit need to exist in the tree? We know they don't go to it when it's on the ground, but what if the tree's not there? What if the fruit's hanging in midair? What if the tree is not green? What if the tree is pink, or purple, or yellow? All of these things are very difficult to do in actual real world, but you can do them in a virtual world. So one of the ways we're studying this question is we're basically presenting our insects with a virtual reality, where we can manipulate their environment in real time. We can change cues, we can change the visual world, we can make a tree pink or green, or we can make it disappear altogether and see how these insects cope with being able to find their objects. So I'll walk you through what our setup actually looks like. So this is what the setup looks like. We have here a high-speed camera, which allows us to visualize the flies in flight. We have a visual display, obviously, that presents the objects, and this is a high refresh rate monitor, about 167 hertz, because as I told you, the flies don't see well, but they see fast. We have a tethered fly. Can you see the little fly? It's white there on the very edge of this syringe needle. So we basically super glue a syringe needle to their back, and that's how we suspend them and keep them from flying away. But we make sure to be very careful not to hinder their ability to fly. So they can't move, but they can still fly. We also have a high-frequency odor, which allows us to stimulate with odor at a very high speed, just as if they were flying and smelling it while in flight. And we have a laminar airflow, because as they're flying, they need to detect odors that are in a wind. So that's how these insects detect odors, is by detecting them in an airflow. So we've got all these different things together. And this is what we do. So this entire system was built by Pawan, who's a graduate student in the lab. And what he does is he takes these tethered flies and he looks at how they beat their wings. So if you've ever been in a rowboat, you know that if you want to turn left, paddle, 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 paddle on your right, and if you want to turn right, you paddle, paddle, paddle on your left. And the flies do the same thing. So if they want to turn left, they beat really, really hard on their right, and if they want to turn right, they beat really, really hard on their left. So by measuring the amplitude of their wings, we can, by default, see where they want to go. Do they want to be turning left, or do they want to be turning right? And then we can move the world in that way. So I'll show you what this looks like. These are just two objects. They're just green and red. They're not apples or trees or anything. They're just two big balls of different colors on the horizon. And we say to the fly, okay, fly, where do you want to go? And they can go wherever they want. They can fly out of the world if they want to. They can fly straight ahead. They can do whatever they want. So we measure their wings. So right now the world is not moving. We give them a set time. And you can see right now the left wing is going to start beating more than the right. You see it kicking up, and that kicking actually turns the world towards this green sphere. So this is how we can see what their intentions are, for lack of a better word. And we can see where they're going to go. And we do this many, many times in many different environments. And at least with these two objects, we see, well, they have a preference for big green balls, which really isn't that informative from a natural perspective, but it does tell us something very important, which is that they think there are objects there. There's nothing really there. This is just a virtual world. This is a computer game. But these flies always go to green no matter where we put it in the world. And so that shows us that they actually do see objects. And we can also give them airflow. And we can give them a global airflow to see, for example, if you're flying upwind or crosswind or even potentially downwind, does that affect your ability to detect these objects? And so you can see right now the airflow is actually moving around, and the insect's moving in the world, and it's still going to go to this green ball. It really likes these giant green balls, whatever that represents. We don't know what it means right now. But we do know that the airflow doesn't affect them. So this setup is what is allowing us to test how these flies find objects. Because we can give them complex environments, we can give them trees, we can give them trees with fruit, without fruit, we can give them fruit without trees, we can give them fruits with smell, without smell, all of these different manipulations from all of the information that I've been telling you about these different systems, and be able to identify how insects find objects and what is an object in the real world. So I just want to, again, I really want to thank this really great group of people that I work with. We're a very diverse group, chemists, we have microbiologists, we have bioinformaticians, we have neuroscientists, we have ethologists, we have computer scientists, and we have engineers, and we all work together, and we constantly challenge each other to ask these big questions that I don't think we're ever going to answer, but it's really the journey that we take that we think is the most important. And here's their names, and I also want to especially thank Karin Nordstrom, who's my collaborator from Uppsala on the Hoverfly project, and Jeff Fader, who's my collaborator at the University of Notre Dame. And I'm going to leave you, before we ask questions, with this quote, which is really the motto of our lab. And I hope that you also will put nature first in your class and in your research. Thank you very much.
Good evening, ladies and gentlemen. It's our fortune to have heard a wonderful talk on tracking the objects of insects affections across species and continents. It has been very informative and remarkable talk, madam. On behalf of Bangalore Science Forum and all of us here, I propose vote of thank you, Dr. Shannon. Tomorrow's talk by Dr. R. Govindarajan, Professor, Chairman, Supercomputer Education and Research Center, Indian Institute of Science, will be speaking on supercomputing, range for ranking or science. All are highly invited. Any questions, please?
I have many questions, but I'll just ask one. And that one is, when you were doing those experiments in Skip, you mentioned earlier that there are more than 6,000 species of hoverflies. Were the species that you were looking at, I won't use the word using, in Sikkim in any way similar to the ones in Uppsala or in Bangalore? They were.
They're the same species, yeah.
Oh, that's interesting.
Now, we don't know the population genetics. So, I mean, I say same species taxonomically and morphologically in the classical definition, but we don't know the genetics yet, so we have to collect them.
keep my other questions for the after.
Okay.
Ma'am, are all the behaviors in insects fixed action patterns?
I'm going to ask you to define what you mean by that before I answer the question.
Like everything runs by instinct and not by intelligence.
What do you mean by intelligence?
Intelligence in the sense they learn after their birth by the environmental conditions, and instinct means they do not learn anything and everything is there when they are born.
Yeah, that's a good question. I just wanted to make sure I knew what you're talking about. So certainly all organisms, even humans, are born with some innate abilities, right? We even have some things that we recognize without having to learn them. Faces, for example, are a good example. We recognize faces innately, but we learn whose face it is from learning. Insects are the same, right? So a butterfly, for example, maybe knows what a flower is, but it will learn which flowers to go to through experience. So it may go to a flower that tastes really nasty, and then its brain will learn, okay, don't go back to that flower. So they use both learning and innate recognition. Obviously, if you're an insect that lives a very short period of time, some insects live for a day, then you don't have a lot of time to learn about your world, right? We live, thousands and thousands of times longer than a fruit fly. So obviously, the experiences that they're going to be able to get are much more limited. And this is why some of these innate recognitions are so important, because they don't have the time, right? They don't have the time to explore the world and learn what everything is. They have to get up and get going pretty fast. So certainly innate recognition is a very important aspect, but for sure they learn. But intelligence, I don't know, we can have a long philosophical discussion on what intelligence is.
I have a question. This might be as a corollary to your studies. Can you study on mosquitoes and develop a very effective repellent?
I have many friends who are working on that. Much of the funding for insect research in the United States right now is going towards mosquitoes for malaria research, which is more than a little ironic since we don't really have malaria in the United States. But I do have a very close friend who's actually Indian. His name is Anand Ray. He's originally from Calcutta. And he is doing exactly what you say, basically the similar principles that I use. So he's doing an excellent job, and he's identified some natural repellents already that he's now trying to test here in India. So be assured, sir, this work is being done.
My question is related to economic entomology. There is one pest of silkworm called fuzzy fly, Exorista surveillance.
Okay.
And we have many control measures to protect the silkworm from this pest, but we are unable to do that in South India. It is one of the major pests of the silkworm, and we advocate the farmers to control this pest through keeping some colored material outside the laying houses. Example, lime powder plus turmeric powder, mix it, it will turn to red color and it will attract the fuzzy fly. So it will not enter inside the laying house.
I see.
But it is attracted towards that color and it will not go inside the laying house and it will not lay eggs on the silkworm.
I see.
One fuzzy fly can lay 300 to 400 eggs on individual silkworms.
Right.
So it is a big loss to the silkworm as well in farmers, and we have to protect the larvae from the fuzzy fly. But we are unable to control this 100%. It is just 15 to 20%. Are there any measures to control the fuzzy fly apart from pheromones or pheromones? Any physical barriers can be done in attracting, like you showed some flowers and all.
Right.
So, can there be any devices to control this fuzzy fly to prevent it to enter into the laying house?
Well, I would suggest you try to understand the ecology of this pest, right? And it sounds like that has been done somewhat. So, but really understand what it is. What is the silk silkworm to them? What is it, right? What is it that they're attracted to, right?
Because they have to continue their life cycle. Each oozy fly will lay 300 to 400 eggs on different larvae.
But they have to find it.
Yeah,
right?
there are physical barriers. We will put nets, just like mosquito nets, we will put nets around the rearing house to prevent the oozy fly to enter. But sometimes they are also intelligent. They will enter through the rearing house through the mulberry leaf.
Yes.
The farmers carry the leaf to feed the larvae, no?
Yes.
Along with that leaf, they will hide inside the leaf and they will go into the rearing house.
So obviously then that there's not really much you can do, right? I mean, it's like if you bring mosquitoes into your house, right? I mean, you can't really, then worry about repelling them outside.
It's a deterrent pest.
There's always some preventative measures that have to be taken. I mean, this is true of the coffee problem as well. There are, of course, measures that can be taken by farmers. But the problem is so big now that even the measures that the farmers are taking are not adequate. So, I mean, besides those obvious things, obviously screening and all these things, trying to keep them from getting in, really trying to figure out what they really like and trying to create some sort of, either alternative host source and coupled with a repellent can be a very nice natural mechanism to repel them.
Can we try with some hyperparasitoids for this oozy fly?
Possibly. I'd have to talk to you, I think, a bit more in
We
private. We'd have to talk about what the measures have been taken.
Host is the leptopteran, pest is the dipteran.
I mean, that's one of the things that I don't think I really mentioned in the talk, but there's 1 million species of insects on this planet. It's the most speciose taxa in the entire world that we have. And each one has its own ecology. Each one has its own, so really to understand it, you really have to understand each organism. And you can't, for example, use the tactics that you would use for a mosquito on a horsefly, or on your pest, or on the coffee borer, because they're different. They each have their own unique ecology. So that can make things very difficult, because then each pest that comes along, you have to not necessarily start from scratch, but you have to start with the knowledge that you may not have the right technique. So.
Okay, thank you, ma'am. We'll have personal discussion.
Thank you, sir.
Effects of insecticides on crops. Effects of insects on the crops, and it's the problem of commerce.
I'm not sure if I understood the question, sir. And he ran away, so I don't know.
He wants to know what are the effects of insecticides on crops.
Insecticides on crops. Well, obviously, it kills the insects, right? But if it's unmitigated usage, you do the same thing that's happening with our antibiotics right now, which you kill off everything but that which cannot be killed, and that's what survives. So this is why, I mean, just like in all things, right? In all things, there's no cure-all. And the idea is, of course, in the end, is not to eradicate any species from this planet, but rather to learn how to live with it so that it doesn't become a problem. One of the problems with crops, actually, is simply that there's a whole lot of it there. So even if you're an insect that doesn't necessarily love corn or rice or wheat or coconuts or whatever it happens to be, if there's just a lot of it there, I mean, they're kind of lazy, right? So if you are at a buffet and there's like tables and tables of gulab jamun, but you really want chocolate cake, but the chocolate cake is way down the end of the hall, you might take gulab jamun, which I actually like, just because it's there. And this is a lot of times why crop pests become pests, because there's just so much of their resource there. And animals are a little lazy, right? They may want something like this ideal thing, but if they can't find it, they'll settle for what they can get. So the idea is, can we make these crops less attractive to them? Can we make the population lower, not eradicate it fully with pesticides, because then you just leave these resistant insects. But can we use pesticides intelligently? I'm not against pesticide use, but I'm for intelligent use of pesticides to lower the population to a point where other ecological methods of repellency and attraction, alternative crops, these traditional practices that existed in India and were so well used prior to the introduction of pesticides, There's a lot of plants that are known as natural repellents, natural things. There's a lot of knowledge here in this country, right, that we have to continue to use along with these pesticides. So I'm sorry I became a bit preachy, but that's a very important topic in my heart. So I think this is a very essential one for this country as well.